So we're almost through with the process business. So translation is the last big process that we talk about in the class. We'll be turning our attention to some structural things, to some metabolic things. And I think as we get into metabolism, especially those of you who are interested in nutrition, for example, we'll find some relations to physiology and so forth there. So if you bear with it, we're getting through the last of the process stuff here. Okay? Yeah. All right. So that has nothing to do with the review session, though. So anybody have any questions? Yes, sir. Sean.
Do you mind going over attenuation one more time?
Okay. Attenuation, the most popular process I talk about the whole term. So of all places for the videotape to fail, it was in the attenuation. I did dig up a lecture that has that in there. But again, I'm happy to go through this here. So let's talk about attenuation. And right here. Okay. All right, so let's think big picture first. Why do cells have attenuation? What's it do? If I said, why is attenuation important, what would you tell me? To do what? Control what? Of what? Right. So it's trying to control how much tryptophan is made. Right? So anytime we think about regulation, anytime we think about control in the cell, the reason cells are doing this isn't just to give students something to memorize on an exam. They're doing this because they're responding to their environment. Responding to their environment. That environment says, hey, I got plenty of tryptophan, I've got no tryptophan. All right? So it's responding to that environment. So that's why cells have regulatory processes. Attenuation is a regulatory process. Okay. Now, attenuation is complicated because it uses translation to control transcription. It's an odd scheme. But as I said, it's not unknown. Most of the amino acid operons in E. coli are regulated by attenuation. Okay? So attenuation uses translation to control transcription. All right. So, first of all, let's not mistake what we see on the screen. What we see on the screen is the product of action of RNA polymerase. RNA polymerase has started transcribing, that is, making the RNA for this operon. It's already started. There's 50 bases from the 5' end, and there's 130 bases. Okay? It's moving, and it's ready to make this entire operon if necessary. So what this attenuation is going to determine is whether or not it's necessary. It's determining whether or not this process is necessary. How does it do that? Okay. Well, the way it does it is it uses the ribosome as a barometer of the amount of tryptophan in the cell. What's a barometer do? It tells us when the pressure is high and low. It tells us that changing environment, right? So the changing environment of high and low here is high levels of tryptophan or low levels of tryptophan, right? That's the barometer that the ribosome is providing. How is the ribosome providing it? Right? It's providing it by the speed with which it operates. The speed with which the ribosome operates. If there's plenty of tryptophan, the ribosome's speed is high. If there's little tryptophan, the ribosome speed is low. Now that's the key to the whole thing right there. If there's something you want to think about the process, think about high, fast, low, slow, right? If the ribosome is moving fast, the ribosome goes shooting through here. It makes it through these tryptophans. No problem. There's plenty of tryptophan. It puts those into a polypeptide and gets all the way over here. When there's plenty of tryptophan, the ribosome is sitting right over here, and these two sequences compare, and these two sequences compare. When it's fast, a terminator forms. This terminator that you've seen before kicks the ribosome, the RNA polymerase off, and transcription stops right there. Instead of making a long RNA that has 10 more genes on it, it's only made an RNA that's got 130 nucleotides on it. Okay? So when tryptophan is abundant, we have premature termination of transcription. That's why I keep transcription, translation separate. Okay? Premature termination of transcription. Yes, ma'am?
So does region 1 determine whether the ribosome was thrown away, or, I mean, not that, but determines like which one pairs?
Okay, so the position of the ribosome really determines whether or not this 1 and 2 and 3 and 4 pair or 2 and 3 pair. Okay? So now the difference is a little hard to see on this one. The difference is right here. If tryptophan is low, the ribosome is sitting here waiting for tryptophan, because this genetic code is saying, I need a tryptophan to put in right here. The ribosome is sitting here waiting. Well, when the ribosome is sitting on this sequence, this guy here can't pair with the blue. It can't do that because the ribosome is covering it up. That allows 2 to pair with 3. And 2 pairing with 3 keeps 3 from pairing with 4, and 3 pairing with 4 is what's necessary to terminate the process. Does that make sense? So really the speed of this guy is determining whether or not termination occurs prematurely or doesn't occur at all, in which case the RNA polymerase goes along and continues making the rest of the operon. Yes?
You said the polymerase was sitting on the area where tryptophan is supposed to come in, right?
No. The RNA, the ribosome is sitting there.
Ribosome. Gotcha.
Right. Because the ribosome is doing the translation. It's complicated. Yeah. It's one of the more complicated processes we talk about. Yes, question?
Yeah, are we done with this?
What's that?
I have a question.
Yeah, any other questions on this before I move on?
So
Yeah.
So then this process can only occur in prokaryotes, right? Because it requires ribosomes.
Very good question. His question is, this can only occur in prokaryotes, correct? Because this transcription and translation occur together only in the prokaryotes, and that's correct.
Is there
All right.
something like analogous in eukaryotes?
There's not something analogous to this in eukaryotes because translation occurs in the cytoplasm and transcription occurs in the nucleus. So no. Yeah. Eukaryotes have a ton of regulatory schemes as well, and some of them are really cool, but we're not going to go into it here. Yeah. Okay, other questions on this? Everybody got it? Okay, as I say, look at big picture on this stuff. It's big picture. What are we trying to do? What's the cell trying to do with this? All right, you had a question.
Are phosphotriacylglycerols and glycerol phospholipids?
Okay, so getting into some nomenclature here. Are glycerol phospholipids and triacylglycerols the same thing? All right. Well, let's go back to our thing here. And so you see triacylglycerols, you see triglycerides, you see glycerophospholipids, you see phosphoglycerides, you see phosphoacylglycerols, and at some point it all becomes Greek, right? All right? Here's where the structure of words really bails you out. All right? If I say phosphoglycerol, what does that tell you? A phosphate, it's got a glycerol, right? If I say phosphoglyceride, what does that tell you? I'll tell you. It's basically the same thing. A glyceride is something that has stuff attached to glycerol. So a phosphoglyceride is a glycerol that has a phosphate and some stuff attached to it. If I say glycerophospholipid, let's put it together. Glycerophospholipid says it's got glycerol, it's got phosphate, it's got lipid, meaning that the things that are attached to glycerol must be lipids. In this case, they're fatty acids. Yeah. So yes, phosphoglyceride, glycerophospholipid, okay, both are the same thing.
And so is that what makes up the bilayer of the membrane?
So glycerophospholipids and phosphoglycerides, same thing, both are components of a lipid bilayer. There's other things in a lipid bilayer. They include sphingolipids, which are different. They include cholesterol. They include proteins. Okay? So all those things will be components of a lipid bilayer, but phosphoglycerides will be part of that, yes. Okay?
So phospholipids and sphingolipids are all found within a single membrane.
So phospholipids and sphingolipids are found in any single membrane in the body, and the answer is basically yes. Okay? Sphingolipids are based on sphingosine. Right? Sphingosine is where the sphingolipids get their name. And though they sort of resemble glycerol phospholipids, they don't contain glycerol. That's the big difference right there. Most of them do not contain phosphate. There's only one that I said contained phosphate, and that was sphingomyelin. Okay?
And so then our glycolipids also found within the membrane.
Okay, so let's think about a glycolipid. So when I say glycolipid, what does that tell you? So glyco tells you sugar. Glyco means sugar. All right? So sugar lipid, meaning this is sugar, it's attached to a lipid. Most commonly the lipid is a fatty acid again. All right? And so examples of those include things that I described like the blood groups, where we have the sugars sticking out. We have the fatty acids stuck in the membrane, and we have the sugar sticking out. That would be an example of a glycolipid. Okay? How about a triacylglycerol? What's that? So tri means three. Okay? Acyl means three acyl groups. Okay? Triacylglycerol means three acyl groups on glycerol. Well, glycerol is 3 carbons, meaning each carbon has an acyl group. An acyl group is a fatty acid. So 3 fatty acids on a glycerol is what makes a fat or an oil, depending upon whether it's liquid at room temperature or solid at room temperature. Okay? A lot of nomenclature, but the pieces of the names should tell you what's in them. Yeah? Okay. Anything else on this? Yeah?
So in order to form a bilayer and not a micelle,
Yep.
we need 2 fatty acids?
Yeah, it's a good question. So why do you form a bilayer instead of a micelle? Okay? A micelle happens with detergents, with soaps, and we can just take sodium fatty acids, okay, and there's only one tail, and they fit nicely into a sphere. When we take something and we put 2 tails onto it and they're physically attached to something else, it's too thick to make a micelle. And so that's why they form a bilayer. Yeah. Okay. I think there was another question on this? Okay, let me get, you had a question on something else. Why does cholesterol widen the TM transition? It's a good question. I don't know the answer to that question. It does do that, and what it probably does is it gives the membrane more of a quasi-fluid character, and that probably is important for a cell. Yeah. Okay, you had a question?
Does an anchored membrane protein project for both sides of a lipid bilayer?
There's an anchored membrane protein, and there were some notes I had somewhere that said yeah, and some notes said no, right?
It should be an integral, right?
Okay.
An anchored protein?
Well, no. So an anchored, first of all, let's talk about what is an anchored protein. So an anchored protein is different from an integral membrane protein. Integral membrane protein, the protein sticks through both sides. An anchored protein may have an anchor that's stuck in the membrane of the cell, but it's got a protein that's attached to it. The protein won't go through, but the anchor could. Okay, that's the confusion.
What's the anchor made of?
Okay, so what's the anchor made of? Typically, the anchor is made of. What would you make an anchor out of if you're going to make an anchor? Put it in the membrane. What would you use? Something hydrophobic, and usually you would use a fatty acid. So cells commonly will use something called myristic acid, which has about 14 carbons long, that is used to anchor things, but there's other things that can be used as well. Yes? Well, I'll come back to you. Go ahead.
Oh, okay. So I was just trying to remember between a nucleotide and a nucleoside.
Yeah.
Does a nucleotide always only have one phosphate?
Okay, so what's the difference between a nucleotide and a nucleoside is your question, and the answer is the difference is a phosphate. So a nucleotide will always have at least one phosphate. A nucleoside will never have a phosphate.
So all the nucleotides that make up the backbone of DNA,
Yeah?
they're only always going to have one phosphate in DNA.
So in the backbone of DNA, the nucleotides will always have one phosphate. That's correct.
But there's other nucleotides that are used for different things.
The starting material is triphosphates because that provides the energy necessary to drive that process. Okay? Okay, you had a question?
Yeah, what was the function of the -35 region? Was it what?
Yeah, what was the function of the -35 region in the promoters, what you're talking about, right?
Just let the RNA polymerase know the promoter and the transition, or
Okay.
the transcription start site is coming up, or what was the?
So our question is really what does the minus 35 region do, and in the promoter, and what is it? How does it help the process to occur, basically? The answer is that the minus 35 provides a good binding site for the sigma factor. The sigma factor recognizes that, it binds to that, and because that minus 35 is not too far away from the minus 10, it facilitates the RNA polymerase getting to the right place to start opening up the strands. Okay? Oh, okay. You're just waving at me. Okay. Hello. Other questions? Yeah?
What's the difference between a nucleotide excision repair and base excision?
Okay. So our question is, what's the difference between nucleotide excision repair and base excision repair? And I went over that fairly quickly, so I'll just briefly tell you the differences between them. As their name suggests, base excision repair involves the removal of a base, and a base only. So we think we got this DNA strand, and there's this damaged base on here. Okay? I don't have to take out the entire nucleotide. I can pull out the base and then replace it with something else. That is a more efficient process. Whereas nucleotide excision repair involves removal of the entire nucleotide and then replacement of that nucleotide. Okay? So I didn't want to get too much into details of that, which is why I didn't go through those, but that's the difference between them. Okay? Yes?
For supercoiling of DNA, does DNA supercoil and then go around histones, or is that two separate phenomena?
That's a very good question also, actually. So the question is, what is the relationship between supercoiling and the wrapping up of DNA around histones? Okay? It's a way more complicated question than I can answer for you right here.
That's okay.
So suffice it to say that the histones facilitate a sort of a self-wrapping of the DNA.
The DNA that's wrapped around the histone is not supercoiled?
Okay, so
I guess that's more so my
okay, so her question is the DNA wrapped around the histones supercoiled or not? In the local region where it exists, no, and supercoiling will tend to favor release of the histones. Okay, but over a longer stretch, if we count the number of turns and bases and so forth, it actually is, yes. Yeah. But it's a very complicated, it's way more complicated question than you probably thought.
Figure out if they were connected. Because we talked about them one after the other. So
Yep. Other questions? You're falling asleep.
I have one more.
Yes, ma'am.
Is RNA polymerase 1, 2, and 3 only in bacteria?
Okay, so our RNA polymerase 1, 2, and 3 only in bacteria. No, they're in eukaryotic cells. So bacteria only have one RNA polymerase, only one. It's eukaryotic cells that have specialized RNA polymerases, yeah. You guys feeling confident about this? Not confident? More material than the first exam? Less material? People always say more, right? Yeah. More new. Okay. Nova?
Can you talk about the difference between transcription factors and enhancers?
The difference between transcription factors and cancers?
And enhancers.
Oh, and enhancers. Okay. That's a good question. So I'm going to give you what I hope is a simplification for that. Okay. So our question is, what's the difference between a transcription factor and an enhancer? We will use the terms interchangeably. Okay. So what are they then? Why do we give them different names? All right. So technically speaking, a transcription factor is a protein that binds the DNA and affects transcription. Okay? It's a protein that binds the DNA and affects transcription. An enhancer is a protein that binds the DNA and affects transcription. It just happens to bind to sites that are very far away from the transcriptional start site. But an enhancer is a type of a transcription factor. Okay? And only eukaryotes have the enhancers. That's right. So sigma factor would be considered a transcription factor because it's binding to a specific sequence on DNA and it's affecting transcription. You bet. Yeah. There are many, many transcription factors. And in eukaryotic cells, I didn't say too much about it. I talked a little bit about how the enhancers give some tissue specificity, and they do that. Okay? But eukaryotes have many, many, many transcription factors. And some of those transcription factors play very important roles in decisions of cells to divide or not divide, and thus can be thought of as oncogenes, because oncogenes are genes that, I should say proto-oncogenes, are genes that can affect cell division and cancer. So we'll talk about those later. I keep promising you guys that. So let me give you a couple tips about studying. I think I've sort of talked about these a little bit before, but in general, I recommend that you start with your highlights, with my highlights, and use the highlights as your guide to studying, just like I use the highlights as my guide to writing my exam. That's how I write the exams. I sat down last night, and I looked at the highlights, and I said, oh boy, I can fool them on this, et cetera, et cetera. No, I didn't do that. But I did use the highlights. Yes, ma'am?
There's a process in the highlights that we didn't talk about in class.
Yeah.
Should we know that?
I do recycle my highlights sometimes, so if I haven't talked about it in class, I don't ask you about it. Yes?
Can you just go with thymine dimers?
Thymine dimers, yeah. So today is a good day to get thymine dimers. Okay? So whenever you're tanning, whenever you're out getting UV light, you are making thymine dimers. And thymine dimers are actually covalent bonds between two thymines on the same chain. So if I have a sequence G, T, T, A, G, and I expose that on one strand, and I expose that to ultraviolet light, the two T's can become covalently linked to each other. Okay? Which is bad because it has to be repaired. If it's not repaired, then the DNA polymerase chokes and has a problem. It can mutate. It can cause a variety of things to happen. It might even cause a break to occur in the DNA. So there's serious things that happen if those are not repaired, and that repair process is very important. Yep. That's why I say you don't want to overload the repair system, because if you do, then you're really potentially asking for trouble. Anyway, so let's see. What else can I tell you? So start with the highlights. I think highlights are important. I find, and it's a little different on these first two exams, but the next two exams, you're going to find flashcards are really going to be your friend. Okay? Flashcards, every piece of information, put it on a flashcard, because a lot of this is just, let's get the language into our head. Let's get the things that we're doing of these processes into our head. And flashcards, I think, really help in that process. The other thing I recommend doing periodically is, if you don't study with somebody else, touch bases with somebody else. Personally, I don't find studying with somebody else usually to be very efficient, because people gab and they talk, and some people are more into it than others are. But I see people looking at each other and smiling. I won't point any fingers here. But suffice it to say that talking to somebody else oftentimes will make you think, oh, I didn't think that was important. I didn't think about such and such. And learning to pick up that information from somebody else is helpful. Okay? If you don't have somebody else that you touch bases with, send me an email, and I will put a listserv out there or something where I can connect people for that. Okay? So it's useful to touch bases with somebody else on studying. The exam is 20% of your grade. So first exam is 20% of your grade. So no one exam is going to kill you, but it's important to do well on the exams as you can. I will be in my office tomorrow. I've got a big meeting in the morning at 9 o'clock. I told somebody earlier I'll probably be in that for close to an hour, I'm thinking. But other than that, I should be in my office most of the day. So I'm always happy to interact with you if you have questions, and go with that. So.
Is there a question from the syllabus?
I'm sorry? Is there a question from the syllabus? I don't know. Should I put one on there? Extra credit. There is an extra credit question on there. There is an extra credit question on there. I can tell you that much. What's that? It's a question that every person in the class, I think, should be able to answer. Okay? So it's not, I'll tell you this, it's not about biochemistry. Okay? But it's about the class. Okay? So that's really mystifying, isn't it? But it's not a syllabus question. So I don't have a syllabus question. I can put one on there real quickly. Okay, folks, study hard. As I say, I'll be around. Give me a holler. Indeed. Yes, sir. Oh, God, you know, I didn't do that again. I apologize. I'm brain dead. I've had a busy week, and I should.
Yeah.
That's not an excuse. I need to get that done. I apologize. I don't have that for you.
Okay.
I've got to get that done tonight. I will get that
All right.
to you. Okay?
Sweet.
Sorry about that.
That's all good.
Brain dead professor.